So welcome to CNSI, everybody, and hopefully this will boot up.
There we go.
Okay, so how many of you in this room have ever taken antibiotics? Ever in your life? Okay, pretty much everybody. Did you get better? Yeah? Okay. Well, you're lucky. And the reason why you're lucky is shown here. So this is the report that came out earlier this year regarding an individual that was infected by something called New Delhi metallo-beta-lactamase producing Klebsiella pneumoniae. And what happened is a 70-year-old female appeared in the emergency room in a hospital in Reno with a very, very serious blood-borne infection with bacteria. She was septic. They cultured out of her blood this organism right here, Klebsiella pneumoniae. But the problem was that it was resistant to every single antibiotic available. She was put on life support. They did their very best. Unfortunately, she died shortly thereafter. And the antibiotics include these are called carbapenems. We save these in the hospital for the really, really bad resistant bugs. It's one of the last resorts. Her strain was resistant. Also resistant to this antibiotic called colistin, used to be used a long time ago, but it's toxic for kidneys and toxic for nerves, so it's been put on the back shelf. But now we use it for these kinds of bacterial infections. And so this is a story that we're seeing repeating over and over with increased frequency. And there are many of us that are very concerned about why this is happening and particularly focused on finding a solution. So the antibiotics you took are likely to have fallen into this class, broad spectrum antibiotics. What that means is they will probably kill the organism even though the doctor might not know what that organism is. So they're broadly effective against many different bacteria. But the problem is that bacteria evolve very quickly. And what they do is they take genes that encode resistance to antibiotics and they string them along in a linear DNA, usually on plasmids, small pieces of DNA that can transfer from one bacterium to another. So they trade antibiotic resistance determinants with each other, and when you take a broad spectrum antibiotic, you're essentially selecting, for example, for your gut microbes to obtain these resistance genes. The other thing they do is they disrupt our healthy bacteria. This is the gastrointestinal tract, and as you all know, you are filled with bacteria, trillions of bacteria that we carry around with us. And by and large, they don't do us harm, and they actually protect us against pathogens. And we take antibiotics, we cause what's called dysbiosis, which disrupts this very important protective barrier that we have. So what's a solution? Well, potential solution, we have the slide thing happening again. Potential solution is shown here. And that is precision antibiotics. And the idea is that we could make an antibiotic that only killed specific pathogens. Nobody else that would eliminate this resistance for broad host range transmissible antibiotic resistance, at the same time prevent this from happening in the gut. So this is a new idea. We don't have any on the market yet that are precision. How do we do that? And one approach is actually to go back to nature. And so how many of the high school students know what a bacteriophage is? Okay, some of you don't. Well, it's really simple. They're viruses that infect bacteria. And in fact, they're the most abundant life forms on the earth. They are extraordinarily diverse. They replicate fast. And these are some bacteriophage attaching to an E. coli. And here you can see the virus is injecting its DNA into the bacterial cell. And the way they do that is they have a structure called a contractile nanotube, since this is the Nanosystems Institute, that recognizes binding to the bacterial cell surface, and that binding triggers deployment of a tube that injects the DNA. The DNA replicates, and outburst hundreds of viral progeny. Okay, so this has been evolving for 3.5 billion years. There were bacteria and bacteriophage long, long before we were ever here, and they will be here long after we're gone. And so if you look at these numbers here, very large number of phage, and there are an estimated 10 to the 23 infection events per second. That's an astronomical number. And out of this evolves efficiency and specificity of killing, and specificity is determined by binding to the bacterial cell surface. Turns out, in nature, there are even more useful particles, and those are shown here. They're sometimes called R-type hyacinths, and what they are essentially nanostructured bacterial killing machines. That's all they do, and they do it exquisitely well. And so they bind to outer membranes, the outer surface of a bacterium, and when they bind, they inject the same tube, but there's no DNA, there's nothing to plug the hole, and so ions leak freely. And leaking protons for bacterium kills the cell, because that's how they respire. And so these kill with single hit, they're highly specific, and we can swap these binding determinants for these binding determinants to change their specificity and to reprogram. So one of the things we're interested in here at CNSI is understanding the biophysics, the molecular biology of how this works. And we want to understand that so we can engineer them to be useful for us in the context that we need them. And so this is an electron micrograph of these particles. This one has not yet fired. This one has, and you can see that tube extending down. And this is a cryo-electron microscope structure made by our collaborator, Hong Zhou, who has an incredible electron microscopy suite in our basement. And the way these are structured is there is a sheath surrounding a tube, and inside the hole is hollow. And that's what leaks those protons and other cations after injection occurs across the bacterial cell surface. The structures are really interesting. And what's happening here is this is before it fires and after it fires, and what you saw is that it got shorter and fatter. And what's actually happening is the sheath subunits that surround the tube are transitioning from a high energy metastable state to a lower energy state where they're very comfortably interacting with each other, and this is the preferred state of the system. And so what this essentially is a biological battery. It's designed to store the energy it needs to puncture the bacterial cell, and it does it in the absence of ATP with no other exogenous source of energy. And that's what's really beautiful about how nature constructs these particles. And so here you're looking at this pre-fired state. The tail fibers don't resolve because they wiggle around, even at temperatures 170 degrees above absolute zero, which is what we use in the cryo-electron microscope. So you don't see them here, but this is really the business part. It recognizes binding to the cell surface, triggers, and the energy that's released by this transition powers the injection of that tube across the surface. Nothing specific required from the bacterium. This is a generic killing mechanism, and it's extremely stable. And if you remember from your high school chemistry, we can look at the energy that's released. And when this happens, there's 12 kilocalories per mole of energy released per sheath subunit, times 162 sheath subunit gives about 2,000 kcals per mole, which is a lot of energy. And so these are able to very quickly and very powerfully kill bacterial cells. Okay, so what are they good for? I'm going to show you just one example because I want to stay on time. A few years ago, in the summer of 2011, there was one of the largest outbreaks of foodborne disease that we've ever seen. And this happened in Europe, and it was all throughout Europe. First, Germany, France, Spain. Spain was initially blamed for it. The Germans said it was Spanish cucumbers. And the Spaniards said, no, it looks like it came from Germany, and they were right. And in fact, it came from sprouts. And sprouts are germinated at 37 degrees, body temperature. in water. And you think they're supposed to be healthy, and they are, as long as they're not contaminated with bacteria. And unfortunately, these were. And so the bacteria caused disease very efficiently. All you needed was about 50 to 100 bacterial cells. That's a very, very small amount of bacteria. You'd never be able to see them unless you're looking under a microscope. What they caused is bloody diarrhea and kidney failure. And the two are related by a very bad toxin. And if you look at the numbers, 4,000 cases, about a quarter of these people had to be hospitalized in the intensive care unit, and morbidity was significant. What happened is apparent from the genome sequences. So what we do as microbiologists is we sequence genomes, and comparing them told us exactly what had happened. This was a new pathogen, never seen before, newly evolved, and a bacteriophage with a really bad Shiga toxin landed in a enteroaggregate of E. coli genome. And that event made a new pathogen, and this probably happened sometime in the late 2000s or so that had never been seen before, and it swept throughout Europe. Okay, so what can you do about it? Well, we went to these contractile nanotubes, and we took some that were made by Pseudomonas, and we looked in these sequences, and what we saw were genes for bacteriophage. And that often happens in bacteria. They insert their genomes into the bacterial chromosome, kind of like HIV inserts its genome into a human T cell chromosome. And so you can look and you say, there's a tail fiber gene from a bacteriophage, there's another one. And so we accessed the sequence, we found prophages, synthesized the putative receptor binding protein genes. We didn't even have the strains isolated. Cloned it into our scaffold, the contractile nanotube, expressed, isolated, and tested, and sure enough, in 2 weeks, we were able to create a new antibiotic that was able to specifically and effectively kill this E. coli O104, and this is a lawn of bacteria. These are dilutions of this contractile nanotube. But what was beautiful about it is it didn't kill any other bacteria tested. And it's possible to make these reagents against many different types of bacterial pathogens. And so the take-home message is that a platform scaffold, sort of a generic killing device that can be reprogrammed by swapping tail fibers, is kind of the basis of this. Okay, in 2 minutes, I want to tell you about something else we work on in the lab, and they're just now starting to come together, and where this is something we're excited about. So this is a group of retro elements that were discovered in my lab about 15 years ago. We've been studying them a long, long time. We now know that they're present throughout the bacterial and archaeal domain of life. This is about 30% of all of the diversity of the tree of life that you're looking at right here. There are thousands of these that have been identified, and they diversify ligand binding domains of bacterial proteins, outer membrane proteins, and sometimes even phage proteins. And the way they work is they have a really unusual structure. They have a reverse transcriptase, a gene encoding a protein with a ligand binding domain, and other components. And what this does is introduces diversity into this binding determinant. And the level of diversity is vast, up to 10 to the 25, 26 variants, much, much more diverse than our immune system is when it tries to make antibodies or T cell receptors. And so if you look at a phage that has a certain type of binding specificity, that's encoded by this region right here. The process involves an RNA transcript, which is reverse transcribed by this gene product as well as an accessory gene. And this is unlike any other enzymatic complex that makes DNA, because what it does is when it's copying its RNA template, it copies a G or a C or a U with no problem. But when it sees an adenine, it throws up its arms, doesn't know what to incorporate, and it mutagenizes at those sites. And so what you get is the same gene, except the ligand binding part has now been changed as a result of this mutagenesis. And when we look and see where the mutations are structurally, they're exactly where where we would want them to be. They're in the ligand binding pocket of the receptor binding protein of that phage. And so what this gives us is a way to very rapidly generate new binding specificities for this novel class of antimicrobial agents, which we hope someday will allow us to overcome, or at least help to overcome, the problems with antibiotic resistance and microbiome dysbiosis that we now face. Okay, my last slide. Anybody know what this is? These are bacteria, and they're all colored different colors, something called multispectral fluorescence in situ hybridization. Anybody want to take a guess? Why you brush your teeth in the morning. This is dental plaque. And just a few of the bacteria, the 400 or so species that build up on the surface of our teeth and our gums overnight. And one of the other applications of this technology is, boy, wouldn't it be nice to be able to take out the bacteria that cause tooth decay? Or wouldn't it be nice to take out species that are responsible for colonization of the ones that give us periodontal disease and gingivitis and other diseases? And these same reagents can be used, we think, to manipulate microbiomes in the oral cavity, in the gut, and potentially other environments. So I'm going to stop there, thank the collaborators that helped us with this work, and turn it over to our next speaker. And thank you all for coming.